Yeah, probably 10 minutes.
Can I go ahead?
Please do.
Okay.
In about 10, 12 minutes. Thank you.
Good evening. I hope everyone, it's evening on my side at least, and good day, everyone. I hope you enjoyed the talk, the workshop so far. I will just quickly rush through my presentation. It was designed to take 20 minutes, but I will try to compress it to 10 minutes. I will see. Just here to report on the livestock genomes references that some of the work that we've been doing so far in South Africa, specifically through UNISA Group, through the partnership with the AfricaBP. There's this project, part of the branch of the project is done under the Nelson Mandela Genomes Initiative for Conservation, with the specific aim to understand the genetic basis of the cultural and economically significant animal and plants. This is done through the effort of building the genomics capabilities within the continent that will serve the purpose of answering some of the questions that we had that include the food security, some of the challenges that we have, such as the biodiversity conservation, biotechnology as well, that can feed also in the medicinal purposes. This is relating mostly into the plants. Some of the work that we have done so far in terms of generating high quality reference genomes, we have managed to sequence at least 20 genomes so far. I was told that this week we can have more addition. They are finalizing some, the number will go up. Among this completed one, we have 7 that only have the PacBio Hi-Fi, and 13 of those have both the PacBio and the Hi-Fi Omni-C. This is just to take you through some of the method that we use. We generally collect fresh samples, can be the leaves or can be fresh blood from the animals that are put into dry ice, then transported to the lab for processing. We previously used to employ the SQL2, PacBio SQL2, and for hi-fi reads, and currently the Ingaba presentation did mention earlier on that we have partnered with them and we purchased the PacBio Review. So all the new genomes that are processed, they are done through the PacBio Review. And for OmniC, we used to employ the NovaSeq 6000, but currently we also making use of the Onzo. These are all the equipment that are available at Ingaba. In terms of some of the part of, in terms of the sequencing I didn't include because Acclaim did good justice on that earlier on. He really touched bases on, like, well detailed on the processes involved in the sequencing. In terms of the genome assembly, after sequencing, we generally employ the genome scope to understand, to do genome profiling, which is followed by assembly done through hi-fi system, then for scaffolding, sometimes with paging, then contamination, then followed by manual curation. This is just to give you the overview schematic of what we do in terms of the genome assembly. So just to report on some of the things that we have done, of the breed that we have done so far, we have sequenced the small stock in terms of the Nguni sheep, which are generally indigenous to Southern Africa region. They are well known to be well adapted to different agricultural regions, and they are the so-called the breed of choice when it comes to the smallholder or poor resource farming systems. And also we did the Nguni goat, which is also indigenous to the South African region. They are generally characterized by small body frame, multicolored and patterned glossy short hair coat. And they also have the exceptional tolerance to harsh environmental conditions. As well, we also did the indigenous pigs, the Coolbrook pig, which is an important domestic species, which is also comprised of a wide selection of commercial and indigenous breeds, regarded as one of the South African indigenous pig breeds, and it's well adapted to the region. This is just to show the characteristic of the or the profile of the genome that we did for Nguni goat and sheep. It is to see the genome size. Mostly of them, they are roughly averaging around 3 gigabytes. You can see the Colebrook is about 2.6, and the Nguni goat is also about 2.8. The Nguni sheep as well, it's about 2.9 gigabytes. So they are averaging around, it's what we expected. And when you look at the other published genomes, so this is just the genome scope on average, as well as the genome scope, the completeness was above 95% completeness. And also this is to show the genome assembly stats for the Nguni sheep. How does it compare with the other published genomes out there? Uh, it's well comparable, and it looks like we did quite a good job. The quality is quite good, and the assembly stats is quite comparable with what it's all out there. This is the Nguni goat as well, and also looking at how does it compare with the others that are out there, and as well as the official genome reference of the Nguni goat. It's very much comparable, and it looks like we actually somehow we did much better compared to what is available there. So we are currently at the right track, and we're producing something of good quality. So also to look at the Colebrook peak, the assembly stats, also we can say we are producing good quality high reference genomes that can also very much comparable to what is already out there. And also just to look at the cattle part, we have done the Nguni cattle, which is comprised of different ecotypes. The Nguni cattle, they're not complete here. We still have 2 ecotypes that we need to sequence. But we did the 3 that we finished now is the Pedi, Venda, and Shangan. They're all Nguni cattle. They are slightly different in terms of some of the phenotypic characteristics. Like there's an interesting study that was done, published recently this year, which compared the weight of the growth traits between the 3 ecotypes, where they reported that on average the paddy sheep, the paddy weaning cattle, they have about the winning weight of above about 156 kg as compared to the 136 and 144 of Venda and Shangan. So all of these cattle, they are well known they have similar characteristics in terms of being adaptable to the environment and well tolerant to some of the problematic diseases that are endemic to the regions. We also included the Bonsmara cattle, the Thuli, as well as the Drakensberger. The Bonsmara cattle is a composite breed which is developed for commercial beef production. It was developed by Professor Bonsma at one of the research station in Limpopo province here in South Africa, back in the 90s, early 90s. That's where it got the name from, because it was developed by Professor Bosma, and it was done at Bosma, and it was done at Mara Research Station. That's where it comes, the name Bosmara there. It is well known. It's one of the most common breed in terms of the commercial beef production setup. They are well suited to a diverse environmental conditions and tolerant to heat and tick-borne diseases. And they are also known for high quality meat. Hence, they are well popular in the setup of commercial beef production. We also did the Thuli cattle, which is a dual-purpose breed originating from Zimbabwe, and it's well adapted to harsh environments. Well, the originating one, uh, officially it says they originate from Zimbabwe, but there are other studies argues they differ. But officially, we will state that it's the good thing. It's indigenous to the South African region, and it's also known for good maternal qualities, as well as the Drakensberger, which is a beef breed native to South Africa, well adapted to harsh conditions, perceived as dual-purpose breed and well tolerant to diseases. Report on the genome profile of the cattle that we did so far. The genome size, it's more of similar, and we expect that they're running around 3 gigabyte, which is something that we see on average, it's around 3 gigabyte. which is something corresponding with the other reported cattle genome sizes out in the NCBI or in other publications. So this is for the Nguni ecotypes as well. They are also ranging around the similar line, and this is the Basco assessment score assessment results, which is also we managed to accomplish at least above 95% completeness. So this is the genome stats, the assembly result for the cattle that we did so far. We are also seeing quite a good results that it's well comparable out there. It's good quality. I wouldn't dip much into this because of time. Also, this is the pretext map that we generated, and we also ran the annotation results. This is the annotation from the Nguni cattle, from the Nguni sheep, I meant so to say, and how it compares to the other three genomes that we with the Rambolite as well as the Hu sheep and the Tibetan and the some of the repeat elements. We also did the repeat elements. I didn't report some of them here. Yeah, and also just to emphasize on the other the one that we have so far, other genomes that we have the indigenous chicken, we have the big blind snake, we have the bush tea, we have bugle brush, the red cormorant, tilapia fish, we have the boer goat, savannah, as well as the Cape speckled. Some of the looking at the benefits, how does this come to play? We have seen the challenges that are associated with crossbreeding efforts, place that the indigenous that put the indigenous species at a vulnerable space, and then we run the risk of losing this good quality trait within the indigenous species. So crossbreeding also we might have good intention when we're doing crossbreeding, but at the other hand, we are running the risk of losing the unique indigenous species. So we need to develop programs that will conserve these precious species. For this purpose, that is the reason why we aim to generate high quality reference genome, which is rather seen as the first step and to realize these efforts. We hoping to emanate whatever the projects that are out there in order to address these issues, and also the potential for future research to unlock more benefits from African indigenous species genomics, and as well as how do we link the bioeconomy. We always advocate wherever we are to establish more collaboration, to work more together. I'm glad that most of the talks today were also emphasizing that we really need to find common grounds in order to collaborate continent-wise in order to realize these opportunities, and also to again call for the stakeholder support in terms of investing in support genomics research for a sustainable bioeconomy. So from the research UNISA research target from this UNISA research team, the target for this year, we are hoping by the end of the year we will at least have 100 genomes completed, and also that should be finalized with both Omnisci and HiFi. We are thinking that we should be on the right track. We are realizing that it's already May, but yeah, we are still optimistic that we will reach the target for 2025. Thank you. I had to rush through time because of